Okay. Good morning, everyone. Welcome to yet another episode of Health Discourse with Sakshi and Sushant. So, hi, Sushant. Good morning.
Good morning.
So, today we are going to talk about a bit about the foods and a bit about how metabolic signals are getting tracked these days and what is the relevance of such, tracking and monitoring, right? So, I think let's just begin with understanding of my first question that Sushant, whenever we see that, foods, they're available both in a package form and in a non-package form, let's say chickpeas, right? And when we talk about their absorption, when we measure the absorption, it is absorbed differently. We have seen that at many instances. And the same is applicable to grains or like oats, right? In a traditional form, you have steel cut oats and all that, but
Mm.
processing makes it a little in a roll form. Yeah, the grain, the whole grain and the roll form, right? And then I can also give you an example of how nuts are processed these days, without the only the kernel is available in the packaging. But when you are taking it along with the shell, let's say walnuts or maybe almonds, when you break the shell and when you eat it, so you find there's a whole lot of the healthy fats, they're still there. But due to the
Mm.
processing, there is some differentiation in the absorption. So my curiosity here is that why is this happening with us these days?
Okay, so let us, since you have brought three examples, let us discuss each one of them, right?
Yeah.
Oats, right? Traditionally, we know oats are really, really beneficial
Mm.
in terms of it do have carbohydrate, but it do also have dietary fiber, right? Resistant starch, and it actually, there are a lot of the way we consume it, right? It gets into our stomach, so there are certain receptors in our small intestine also, right? But they're not strong enough, right? And a lot of these, the density of the receptors on these receptors are actually indicating the, it indicates the brain about your fullness or your hunger, right? And a lot of the density of these receptors and hormones, right, are very, very dense in large intestine, right? So a lot of, we know a lot of this fiber is not metabolized uh, uh, by human body. It is metabolized by the microbiome. So for microbes living in the gut, it's a party for them, right? So they metabolize fiber and synthesize them into various beneficial byproducts, which do regulate satiety, do regulate our blood sugar level. The best example is steel cut versus rolled oats, right? So we know rolled oats is more like a processed form, and steel cut is more of a traditional, right? So there is obviously one factor is obviously there is a difference in the glycemic response to each of these. Besides, there's further impact of glycemic response based on every individual's biology, right? It is not, you and me would not absorb the same food in the same way, right? We would absorb differently, and the calories coming from those foods would be entirely different, right? For you and for me, right? The fiber, how it is metabolized, nutrition again depends upon the functions of various microbiomes. For instance, oats could be used by certain microbes to produce bitrate, right? Again, bitrate we know is an essential short chain fatty acid which central the gut lining, which regulates your satiety, which regulates your blood sugar response, a lot of these things, right? When it comes to roll dose, probably the sugar level, the glycemic spike would be entirely different, right? Would be more than that caused by steel hood in a general phenomenon. But again, for everyone individual, it would be very, very different, right? Uh, so that's why, another thing when people are into weight loss and they count calories, right? It's an, very wrong way of, you know, uh, calculating the nutritional value, because how it is absorbed determines how much calorie you extract from a particular food or how much calorie gets excreted, right? There's need to understand our unique biology, our biochemical individuality, right? It's very, very different apart from the way the food is processed, right? It has a bearing on your metabolic health, on the the metabolites produced by the microbiome will directly impact your physiology, right? So one example is oats, right? Second, obviously, is chickpeas. Again, chickpeas is sort of lentil, right? And it comes under the lentil category, and then it is much higher in protein as compared to oats, right? And again, for people who are vegetarian, right? I just saw someone posting on LinkedIn that how do we get protein for vegetarians? Again, there are a number of ways you get protein, and again, that's a different discussion. I think we discussed about protein, right? That it is not based on population. It is again based on what particular your gene expression profile, your microbiome profile, your function by your microbiome, and what path is active, multiple things, right? A standard quantity of this much protein you need to take is may or may not hold true, right? So again, chickpeas, how it is processed has a bearing, right? So if you take raw chickpeas from suppose a flour mill sort of shop, which gives all this sell all this millets and everything, right? Which is raw form, right? So it would a lot of these would not be absorbed inside your small intestine, and it would land up in large intestine, and it chickpeas do produces byproduct, right? Short chain fatty acid. But again, if it's packaged form, it's processed differently. It may devoid you from certain nutrients, right? Again, it is also about package and also the genetic modification which is happening right now. We are putting a lot of you know herbicides, pesticides, spraying a lot of these things, which is making the reducing the nutritional value of a food, right? What you found on the label is entirely different from what you would actually, consume and absorb, right? So again, there are multiple factors. So obviously, other is the way it is produced, the genetic modification, the agriculture practices, the farm
Yeah, that
practices.
is hybrid seeds these days they are coming, and
Yeah.
what they do is it's a hybrid of more than two, three species, and it actually sort of disturbs. I mean, science
Yeah.
has definitely given us some benefit, but on the contrary, it has also created disasters for us because earlier they used to think if they create, genetic modification in seeds, so their shelf life and the way actually the whole point was to increase the shelf life, and I
No,
think with this we come to the next question also, that Sushant, uh, when we are increasing the shelf life of foods, let's say even milk or packaged grains, like you're talking in packaged lentils, so the shelf life will increase, right? That is for the business and commercial purpose. But what about human life?
okay, so there are a lot of toxic chemicals which are put not only in the agricultural practices but also the way it is stored in supermarkets and on a lot of these shops, basically in metros and towns, right? We are less exposed to these things because we live here in Himachal, but you know, these toxins do activate certain pro-inflammatory pathways, right? It do lead to feed certain microbes doing pathogenic activities, right? So it also makes sure that so there's certain the certain nutrients which even you're taking from various foods are not made available to these microbes. So obviously you do have hunger pangs. You do there is no signaling happening. I mentioned about the hormones which directly interact with the brain to tell whether you are full or you're hungry, right? And it all depends upon what you feed them with, right? Like we take example of oats, we take example of chickpeas, right? Similarly, if you're not feeding those beneficial microbes producing bitrate, acetate, propionate, or triggering stimulation of GLP-1, PYY, a lot of these beneficial, things, it is actually going to even impact your hunger. That's on the metabolic side, but obviously toxicity do trigger a lot of pro-inflammatory pathways. It can trigger multiple issues. For instance, I think last 2 weeks back when we went to Delhi, I had this. we ordered this thing from Blinkit, and these grapes, and immediately after I came back, I had a bout of 3 days of bad digestive health. I had diarrhea, and what we figured out was later on, right? That it was sulfur toxicity, and then there is news around the vineyards and everywhere where grapes are produced, right? There's a lot of this sulfur toxicity being used to protect it from fungi, right?
Sulfur is
Again,
used with most of the fruits which are, perishable to protect
yeah.
from
Yeah.
fungus. So
Right.
that
So
is
that
the
is
point.
one thing, and again, I'm sure whether these supermarkets also putting something, right. So I had bad 3 days full of diarrhea, and I threw up, and
I mean,
then
surprisingly, you were not impacted by anything which you ate outside. It was actually the fruit which caused this. I mean,
yeah,
that is
yeah.
surprising. So,
And for people, a lot of people would go for antibiotics and drugs, right? Which we also bought just for safety check, but I didn't took, right? And for everyone, I just cured it with food, right? With poppy seeds, with a lot of ellagic acid. I took a lot of pomegranate yogurt, fermented food, right? Which
and
helped
so
me.
fennel is something
Yeah,
that was also used.
yeah,
So,
yeah. So these things help me cure, get rid of these things in 3 days, right? And that's so again, this is just an example of the toxicity and the external things being sprayed or being added to the food to change, to increase the shelf life.
shelf
That's
life.
that's a financial model, right? Similarly for nuts, right? You mentioned about nuts, so I'll share about nuts also, right? So every food have these millions of cells, right? The if you see the cell structure, right? It is very, very rigid, right? So, and a lot of these cells, our small intestine enzymes are not strong enough to actually, break these cells, right? And then it goes to a large intestine where a lot of the whole nuts which we consume, it feeds your microbiome. You get a lot of healthy fats like vitamin E, like polyunsaturated fats, monounsaturated fats, which are really, really beneficial for your health, like omega-3 fatty acids, right? So all it doesn't mean you're eating, you're getting them, right? It depends upon how it is being processed, how it is being metabolized by your microbiome, right? So it's these things are really, really key to understand, right? And even, the microbes in the mouth also interact with a lot of these things, right? For instance, cruciferous vegetables like broccoli or cabbage, right? They do have an impact on your bad breath, on because certain microbes could consume and produce oral S2S, which actually have increase your risk of bad breath and increase risk of halitosis, right? So again, you have to see not, just reading the label is not going to help. We have to first understand, how these things are being metabolized inside the body, right? So these things are really, really, really important to understand, right? Coming to that, the blood sugar response again is very, very unique, and I feel I don't buy this point of putting continuous glucose monitoring machines, and tracking your blood glucose level.
Yes, now I
It's
think
it's just waste of money. It's just waste of money.
with this we are actually coming to the next question. Right. Now, CGMs is something that everybody, we have talked about it. We have talked about how it is not going to help you. In our previous talks, at least two or three talks, we have talked about it,
Mhm.
continuously. Now,
Mhm.
here my question to you is that the glycemic response of food, every food that you're consuming, it will change with the time, right?
Mhm.
It is not going to be steady always,
Mhm.
right?
So before that, I'll probably share this thing. I did a video 2 days back on this, but I'll share this thing. Uh, it is important to know in a very layman language so that everyone understands, right? When we put fork inside our mouth, right? There are a lot of biochemical activities happen inside the mouth. So the microbes in the mouth pre-digest these food substrates, and even send biochemical signals to pancreas. For example, when we're talking of the blood sugar response, I'm talking about the pancreas, right? When you take a high carbohydrate meal or foods which are or even foods which are high in sugar or glucose, right? So it sends the signals to pancreas, and pancreas start releasing insulin, right? And insulin generally binds to insulin receptor mostly in the adipose cells, muscle cells, and liver cells, right? So it activates a certain protein called AMP kinase, right? And it sends a signal to nitric oxide synthase, which is lining everything, which is coupled with the endothelial cells, and the endothelial cells is lining with the blood vessel, right? So that signal, it's in the signals, and it to release, it tells the universe to release GLUT4. Again, GLUT4 is a protein which binds to cell membrane, removes the glucose from the circulation and transport it to cells so that, it is being used for it has energy for various activities, right? Daily activities. But for that GLUT4 to be activated, there has to be a signal. That signal is nitric oxide, right? So if you, so either we know oral microbiome or the endothelial cell produces nitrosamine amino acids. If there's a lot of inflammation caused by that microbiome, probably you're eating wrong foods, or your toxicity, or probably foods not married with your biology, and or probably you are using a lot of mouthwashes, fluoride in toothpaste, or foods which are reducing your oral pH level, making it more acidic, right? It harms your ability to produce nitric oxide via nitrate pathway. Again, that is another reason when you have this, your glucose is not clear up the circulation, it goes a message to you. There's hyperglycemia, it goes to message uh, to your pancreas. There is more insulin than you have hyperinsulinemia, hyperglycemia, and that's a cause of type 2 diabetes. Now coming on to continuous glucose monitoring machines. So what we do is, so there are multiple structural and functional issues here, right? So one is, you see, as Indians, right? We do not, say that okay, if I'm having a meal which has a protein, a carb, fat, right, fiber, everything, right? We don't say that, okay, I'm having rice, kidney beans, curd, maybe some vegetables. I don't take, okay, first I'll eat rice first, I'll eat curd, then I'll eat kidney beans and I eat vegetables, blah, 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 right? It doesn't happen like that, right? You mix and eat, right? So the glycemic response is for a meal, right? Not for a food as such, right? First thing. So this doesn't decode the root, doesn't tell you whether a particular food is causing. Again, food is again a sack of various molecules. Now, which specific molecule in a particular food is causing that spike, right? First thing. So there's a structural issue in terms of understanding the glycemic response to various foods. First of all, we focus on meal, we do not focus on food. Within the food, we do not focus on a particular molecule. Right. So again, when you're using CDMs, right, you use for 2 weeks, 3 weeks, and now you know, okay, this is the food is going to spike more, and it's it's like guesswork. You are consuming, and then you're seeing the sugar level spike. You follow for 2 to 3 weeks, and then you know, okay, this food is going to spike more, this food is going to, and you're following again. As you eat, your microbiome composition changes, so the metabolic pathways changes. So a food which was spiking. Sugar much lesser right now may spike more after 3 months or after a month. Again, you will again put that, and you will say, okay, my glycemic response have changed, right? So every month, every 2 months, it is changing. So it's like guesswork, and you'll be, what I have to do. It's why it has changed, right? So the question you should be asking, not, so again, blood sugar response, it spikes if you have nitric oxide, right? Nitric oxide production, it clears off the circulation, right? So so the question is what we are eating, which is disrupting nitric oxide production. What we are eating, which is actually reducing the nitrate production or improving the LPS. So let me show you something how we at Gene Freeletics do. So rather than glucose, we focus on the insulin response, right? So let me show. It is for everyone, and probably people should know this thing. So we have this prediction model where we predict the onset and progression of diabetes. Right? These are the pathways. which is being activated by your gut microbiome. I'm talking the gut microbiome right now, right? It is partly activated by the gut microbiome, right? And this is based on specific molecules these specific microbes interact with, and they express certain genes, which leads to expression of these pathways, right? So we could look at what is the level of activity of each pathway, right? For instance, if bitrate is low, it would increase the glycemic response. Similarly, tyramine is again, tyrosine, tyramine, again, it suppresses GLP-1, it improves your blood, it increases your blood sugar response, right? It spikes your blood sugar level. Similarly, for GLP-6, there are multiple. We look at 17 pathways to determine, combine, and determine the risk of type 2 diabetes. Now, this is not the only thing, right? When we talk of post-prandial glycemic response, right? So we, based on these 17 pathways, we look at how a food would respond to each of these pathway, and then we combine and determine whether a food is going to spike your insulin or not before you consume it. Right? So this is based on your biochemical individuality. So we could know probably if I see high spike, right? I know these things are going to spike high artificial sweeteners, other spirits, beer, sandwich, burger, right? That let me also see medium spikes, right? Uh, in certain cases, this is again up for a particular customer, right? Lemongrass tea may have a medium spike, oolong tea. Again, you won't believe this because you are just seeing the what is going on in your blood, right? That's what you're tracking to CGM. But what happens before that? What kind of pathways being activated, which causes the high glucose in the bloodstream, right? So again, we can look at a lot of these foods which causes like wheat semolina can cause kodo millet could cause medium spike for a particular individual. Maybe for you, may not be for me, right? Depending. Again, these are regulars, right? So brown rice is regular, pearl millets are regular. These are some of the ways we look at insulin response. So CGM's right question you should ask is, what is making what is causing that my glucose level not to clear of the circulation, what is disrupting my nitric oxide production, right? Because again, CGM is guesswork. You're doing 2 to 3 weeks after you again your microbiome changes, your response to food changes, right? So you may be happy. Okay, I'm happy. My I know what is my glycemic response to various meals, right? Right. So again, it will change, and again, it's again after eating you get to know, right? And after a month, again your glycemic response to food is again you'll eat, and so it's a guesswork. You're putting your body at risk, at a metabolic risk.
That is rather interesting because this prediction model and the how the glycemic index is measured. Would you like to shed some light on how this how I mean on what is the background of creating all these? I mean, what is the references that we have taken for creating this?
So there are a lot of existing scientific literature, what we have, right, which have been used to study or define all these pathways, right, with at a molecule level, right, and we look at the gut microbiome. For instance, example of chickpeas, right? If you're taking chickpeas, you have certain microbes like Echinacea municipilia or other microbes, right? Which have a potential to produce bitrate, right? But is it really producing bitrate or not? It all depends upon whether this sense the particular substrate in the chickpeas and produces bitrate, right? So again, we look at how they're interacting with the food molecules, right? Unlike a lot of other testing companies, they look at potential. So they just said you have a bacteria, whether it actually producing bite or not, that is important. So we look at the actual secretion of a certain metabolite, right, by a microbiome based on what genes they are expressing, right? So essentially, it's very, and again, our model is being fed by every single customer contributes to it, their data contributes to it. So again, it becomes more and more precise and accurate with more and more data points. Right?
So you mean to say that for every individual there is a unique set of recommendations which we have already said, and then there is a unique response to each food that has been recommended,
Yes. So,
right?
so for every customer, right? A small bit of my change in the microbial activities would change their pathways. Every customer would have a different level of we quantify, we measure and quantify these pathways, right? So we look at what is the score of each pathway, what is the activity level of each pathway, right? For every individual, it is different. Again, this is mapped with the various entire food substrate and ontology, which help us determine whether a food is going to spike more or less, right? And again, it is based on their interaction with a particular molecule in a particular food, right? It's all combined, right? So one pathway would have, for instance, if someone is consuming too much of, tyrosine, it would lead to precursor production, which could suppress your GLP-1. But again, what level of the activity of each microbe which is producing precursor, plus that particular food may also have certain fiber which would further upgrade byproduct. So again, there are multiple. We look at multiple things here and how different foods. We're taking two different foods together, how it is going to impact your insulin response, right? So the multiple things we look at, right? And that's why it is very, very precise, right? It is, why I have a high response to this food, why I don't have, right? If I have a low nitric oxide production, why I have, how I can stimulate nitric oxide production, right? How I can reduce LPS production, how I can reduce TMA production, right? We look at multiple pathways at a molecular level, right? Not on a plasma level.
So I think with this, we have come to the end of this discussion, and with the kind of insights that you have given us, I think not only diabetics, even people who are in their normal lives, they experience some sort of fluctuations in health. It could be minor fluctuations. It is not necessarily supposed to be a chronic disease, right? So
Hmm.
such information is actually going to help them. What do you think?
Hmm. Hmm. Hmm.
Right? So any last words, Sushant, would you like to add here?
So, I believe everyone's biology is unique, right? Understand your biology, right? Understand because the 99% of the biological processes inside the body are done by the microbiome. Containers, you understand the function of the microbiome, function of the microbiome, not which microbe is there. I think I wrote a blog also on this that it doesn't matter which microbe is there. It matters what a particular microbe is doing, what biochemical activity is doing, what biochemical reactions they are doing, what molecule they are producing. Right? Uh, so it all depends on that. Otherwise any CGMs and all are not going to help you. It is good for. I'll say it's good. It's a good fun toy, right? To check your blood sugar response, but you have to play with this toy every month, and you have to
And
spend.
then you have to, spend money to change that.
It doesn't help,
This,
It doesn't
yeah.
help. So every month your glycemic response would change. So every month you have to spend that money. It's a financial model for all these companies, right? You tell me the cases where using CGMs you have reverse diabetes, and reverse
Mm.
diabetes doesn't mean
No,
getting off
what
medicine.
Sushant, what I feel is that it can only tell you, okay, this is the spike, but it will not tell you what to do. It is not giving you any steps that what you should do. You
No,
except
so
for
so so you know what people would do is okay, spike to a
avoid.
meal, they would start avoiding it, and again,
Avoid.
they
That is
they
the only thing.
they don't know that when they're avoiding, they're also rewarding their body with certain nutrients, other nutrients which are
Nutrients, yeah.
beneficial. And again,
Yeah. So,
after a month, again that food would not raise your sugar level that much, but you
but
don't
you're
know.
not eating that, so how you will know that?
Yeah.
Yeah, na, you.
No, again,
So,
you spend another 5,000 rupees to buy that CGM, whatever they buy, and they fix it every month. You pay, and you buy that strips, and again,
mm,
you would say, okay, this has now not spiking. Again, you have to check your recommendations. Your recommendation would change, and now you'll be surprised. Okay, now what I have to do? Last month there was something else. Now this is something else. Last month, chickpeas was good for me. Now it is bad, right? Almonds was bad for me, and now it is more bad or it is better, right? So again your guesswork, right? So understand the root cause. Start asking the right question. Why I have this, right? Why I have a low NO production? Why I have a high ADPS, right? Why I have a low bite rate, right? So probably why my response to chickpeas is spikes my blood sugar level more than that of my wife or my friend or my kid, right? So again, or my brother, or my father. So again, all these things has to be understood, right? Uh, uh, CGM is not the. I'll say, just a way. If you want to end us in a way that I want to just manage my disease, then probably it's go ahead. It's a good toy to have.
Apparently, yes. So I think thank you, Sushant, for such intuitive thoughts that you have given us, such points for regarding the food and the monitoring of such foods, right, for our bodies. Now, for all the listeners, I would really like to you to share our talks with your friends so that they can also benefit. And we are available on Spotify, Amazon Music, and YouTube. Thank you.
Thank you.